All right, so we're on. This is all about the cell, and like I said to you before, it's not a cell biology class. So first thing I'm going to do is call up a whiteboard and give you an idea of what a cell is, what it looks like, and the constituents of it. So the main three parts to a cell are the cell, the plasma membrane, the cytoplasm, and the nucleus. So if I draw just a generic cell here, anywhere in the human body, the plasma membrane is obviously the outer boundary of the cell. It separates it from the external environment, from the internal environment. And the nucleus, typically, that's what houses the DNA, the genetic information. It's in every cell in the human body except for red blood cells. And when you take A&P2, you'll discover why that is. And then the cytoplasm is just everything else outside of the nucleus, but still inside of the cell. Those are the three main constituents. Now we need to go through bit by bit. I'm just writing plasma membrane. That, as I said, is the boundary between the inside and the interior and exterior of the cell. Plants have a cell wall. We don't call the cell wall in living in animals. It's called a plasma membrane. It's much more fluid and pliable than an actual plant cell wall. The first thing that you should know is what makes up the plasma membrane of our cells, and it's a I'll talk about this a little bit, a phospholipid, which should sound somewhat familiar, bilayer. So we know what a phospholipid is from chapter 2. That's just a phosphate molecule that is polarized and two fatty acid chains hanging down like this. It looks like a balloon with two strings. So this part, the fatty acid chain part, is non-polarized. This part up here, the phosphate, is polarized, and that will be significant in a minute. So that's what phospholipid means. It's just this structure here. Bilayer obviously means two layers, but they're not stacked up on top of each other like this. The membrane actually looks like this because the phosphates are polarized and the lipids, the fatty acid chains, are not polarized. The lipids, the fatty acids, are attracted to each other, and the phosphates are sort of repelled from each other. So the membrane actually looks like this, a mirror image of itself. So that's why we call it a bilayer, because there's two layers of these phospholipids with their fatty acid chains lining up against each other. What this creates is a hydrophobic area in here. So things that are water soluble can't really get through to the other side. Things that are lipid soluble or hydrophobic, think, oil and water don't mix, hydrophobic things can slide right through this membrane, no problem. So in my mind, what I always say to myself is water soluble things, and they tend to be polarized themselves, they can't really get through. It's not that they don't ever get through there, they need help getting through there. But lipid soluble things, fat soluble things tend to slide right through. So that's first and foremost, that's the structure of the plasma membrane. And this arrangement goes all the way around the cell, that phospholipid bilayer. Now, this bilayer doesn't just have phospholipids in it. It's got proteins, cholesterol, carbohydrates, all sorts of things stuck to it and stuck in it. This term, fluid mosaic model, is what we use to describe this plasma membrane. We call it fluid because it's pliable and it's ever-changing. I'll describe that in a minute a little bit more. We call it mosaic because there's all sorts of things in it. It's not just made up of one molecule or two molecules. It's got all different types of molecules in it. So I'm going to draw a quick plasma membrane again. It's easier if I just do this. And then I'm going to stick proteins in there. Here's a protein sticking off sort of off the top. And then I'm going to draw another protein that goes all the way through the membrane. And there are other things besides proteins in this fluid mosaic model. There's cholesterol stuck down in here in different places, usually between these. Cholesterol is fat-based, as you know, it's a lipid, so it tends to be attracted to the other lipid part. So there's some cholesterol. There's carbohydrates that stick off the proteins like antenna, kind of like this. Carbohydrates / sugars. Sometimes there's glyco means sugar. So these would be glycoproteins that I've drawn. And then glycolipids, if there's carbs attached to these molecules of cholesterol. That's really what the cell plasma membrane looks like. It's made mostly of this phospholipid bilayer. And then it's got proteins, different types, stuck in it or stuck to the top of it, the external surface, as well as some cholesterol and carbohydrates. I'm going to say a few things about these proteins. They're called membrane proteins because they're stuck in, I'm just layering over the top of this. I'll stick with blue for proteins. There's basically two types of proteins in the plasma membrane. Peripheral proteins and integral. That's an E. Integral proteins. Just think of this. They're either sort of stuck to the top like this one, and they do not penetrate all the way through the membrane. That's the peripheral. An integral protein penetrates all the way through that phospholipid bilayer. These guys, the peripherals, they can move around in like a ship at sea. They can move around this sea of phospholipids. The integral ones tend to stay put more. They're more structurally anchored. So let's go back to here. Now these proteins have several functions. So depending on the type, I'm just going to generically write out a list of functions of both of these proteins. They can be used for transport. What does that mean? Well, if something needs to get from one side of the membrane to the other, and that something, whatever it is, does not easily slide right through this membrane, then it may use particularly an integral protein to make its way through. Now these integral proteins, they can be channels that are just basically hollow tubes that allow a substance to slip right through this membrane, or they can be carrier proteins. And a carrier protein goes all the way through, it's integral. And let's just say this molecule out here that's red needs to use this protein to get from the outside to the inside. Like I said, a channel protein, that thing just falls right through the tube. But if it's called a carrier protein, this thing binds to something inside of the protein and then gets carried all the way through this protein and then sort of spit out on the other side. So they can be used for, proteins can be used for transport. Let me go back to blue. Receptors. So they can be used for signal transduction. So a membrane protein that's exposed to the outside of the cell may have a binding site for something to signal inside of the cell. So I'm going to say may bind to a messenger, a signal messenger, and then cause metabolic changes. That's kind of a loaded word. I'll describe what I mean. So, for example, if I have a cell here and there's a hormone, their hormones are small. There's a hormone out here, and let's say that hormone is water soluble. It can't just slide right through that phospholipid bilayer. Sometimes there are protein receptors on the surface of cells like that this hormone, because it can't go through the membrane itself, the hormone binds to this protein receptor. There it is. And it causes then, this receptor then is linked to things on the inside of the cell, and it signals the cell to do something, change its chemical reaction. So that's what receptor can mean. That proteins can also act as structural elements. So the cytoskeleton of a cell just means the internal framework of the cell. And that's made up of things like microfilaments, microtubules. Those are all protein-based things. And surface proteins, plasma membrane proteins, can interact with that scaffolding to play a role in the extracellular matrix or intracellular matrix. So we've got transport, they can act as receptors, structure. Some of these plasma membrane proteins can be enzymes. We already know what those are. Those are proteins that speed up chemical reactions. And finally, your book calls this, they're called linkage proteins, but your book uses the word intercellular joining. So I'll write that out. Sometimes they're called linkage or linking proteins. That just means connections from one cell to another. So these surface proteins, I'm going to draw two of them right here on this cell. If they join two cells together like this, we call them linking or intercellular joining proteins. And finally, cell recognition. So these surface proteins or these plasma membrane proteins can also help the cell recognize things on the outside of the cell. All right, the term glycocalyx just refers to the sugar coating on top of a cell, on the outer shell of the plasma membrane. So if I go all the way back to my first diagram, these green things that I've drawn, these green antennae, are starches, they're carbohydrates, and they sort of create this sugary coating, carb coating on the outside of the cell. That's really what the glycocalyx refers to. It refers to this network of carbohydrate molecules on the outside. All right, I'm just trying to call an old board. I believe I left one blank. So there's different types of cellular junctions as well. So, and they all take place within the plasma membrane, and this is from cell to cell. Tight junctions, just like the name says, create a very, very tight connection from cell to cell. And it's basically an impermeable junction, and it's very tight and very sound. It almost acts like a zipper all the way down the cell itself. So tight junctions, or a better description would be if you sewed two pieces of material together very tightly. That's what a tight junction acts like, behaves like, and looks like. Desmosomes are not quite as impermeable as tight junctions. These are more anchoring junctions, and I think the analogy now is like Velcro from cell to cell. And the third main type are gap junctions, which are the loosest type of cellular cell-to-cell connections. Gap junctions allow for some sort of communication to go on between cells, usually the flow of materials. So a gap junction might look something like this. It's not just a connection point, it's a communication port as well. So if I have two cells, and here's proteins creating gap junctions, that allows for the flow of some kind of material or ion, for example, from cell to cell. That would be an example of a gap junction. All right, the next stuff, I'm not gonna beat around the bush. It's not super exciting. The properties of diffusion, simple diffusion, facilitated osmosis, and all that business. So I'll try to be as light and as quick as possible so as to not put you guys to sleep. I have to be honest with you, it's not my favorite bit of lecturing as well. But we're just writing membrane transport. So this topic is about getting something or some things from one side of the plasma membrane to the other, either from out to in or from in to out. And there's different types of transport. There's two major categories. There's passive transport and active transport. When I see these two words, I always think passive means no energy is required. Active means we have to use energy in order for it to happen. Use basically ATP. So passive transport is just basically things moving from where they are of higher concentration to lower concentration. I'm going to write no energy required. Classic example of passive transport or diffusion, which is a type of passive transport. So I'm going to write diffusion. That word just means the passive movement of molecules from a place of higher concentration to lower. And there's no energy required for this to happen. If you walk into an empty room and you uncork a bottle of perfume, eventually the molecules of that perfume will permeate through the air, all corners of that room, but it takes a while to happen. And those are actually molecules of, gas molecules of that perfume diffusing from the area of greatest concentration, which would be in the bottle, to everywhere where it's lower concentration, in the air of the room. That's a classic example of what diffusion is, and it requires no energy whatsoever. Active transport, on the other hand, requires energy. We have to spend some energy for it to happen. And that energy that we spend is that ATP. So our cells make ATP all the time, all day long. But we also use that ATP, that fuel, to perform body functions. And once that ATP, once that terminal phosphate is cut off, that molecule is considered used. Now, there's different ways to regenerate it and to make more ATP, but we have to use those ATP molecules in order to perform this type of transport. This, we say, is moving something against its concentration gradient. Some books call that moving uphill. So rather than, in my example with the perfume, uncorking the bottle and it just diffuses passively from the bottle all throughout the room, those gas molecules do. If we tried to capture those perfume gas molecules in the air and stuff them back into the bottle, that would require a lot of energy. And so that's an example of active transport. There's two different types of passive transport, by the way. I'm just going to mention what they are. There's something called simple diffusion and facilitated diffusion. This is really simple, no pun intended. Simple diffusion just means this. If something, I'm drawing a membrane, simple diffusion occurs if something just slips right through here. Bang, there it goes. Just literally slides right through that phospholipid bilayer. Simple diffusion. Facilitated diffusion is still passive. That means it still requires no energy, but we just need some sort of mechanism like a protein channel for it to get through. So facilitated diffusion, here's our protein, and let's just say it's a channel protein, and now this thing just uses that pore to get right through. That's the only difference between simple and diffusion. Those are both passive mechanisms. Active transport, I'm just going to layer over this. So I'm just going to continue talking about active. Remember, first and foremost, it uses ATP, it uses energy to occur, and typically it requires what we call an ATP pump on the surface or on the plasma membrane. Most people don't really know this, but almost all of the cells in the human body, in our bodies, have these pumps, these ATP pumps, peppered all along the surface of the membrane. So this is a cell, and I'm just going to use a green color. There's an ATP pump, and they're located on the plasma membrane, and their sole job is to push certain things in and out of the cell against their will, against what they want to do. So what that means is, let's just say I have sodium ions, for example, a lot of sodium ions out here. That's Na+. If there's a lot of sodium ions outside of the cell and not so many inside a cell, by simple diffusion, they just want to go in because they want to flow down their concentration gradient, just like the perfume does. What this ATP pump does is it captures that sodium that has run down its concentration gradient and it pumps it back out. That requires ATP to do that. And a lot of these pumps on the surface of our cells are called the sodium potassium pumps. This is just an example of a classic ATP driven pump on our cell plasma membranes. So at the same time that this pump is pushing sodium out, pumping sodium out, it's also pumping potassium in. The only thing, we'll come back to this pump when we talk about the nervous system. This is how a nerve cell sets up the potential for it to fire an electrical impulse, is by this ATP driven pump. The last thing I'm going to say is these pumps they're driven by an enzyme called ATPase. That enzyme actually cleaves or chops off that terminal phosphate to spend the energy in ATP. So that's a classic example of active transport, the sodium potassium pump. It uses ATP and therefore energy to pump sodium out of a cell and potassium into the cell. And it works 24 hours a day. While you're sleeping, it just keeps chugging right along. Okay, so let's talk about, I'm not going in perfect order here of your textbook. I just looked down and saw the topic osmosis. This is just the diffusion of water. Here's a definition of it. Diffusion of, I'm going to say water. It can be any water-based solvent or liquid, but diffusion of water across a semi-permeable membrane. The plasma membranes on our cells are semi-permeable. That means they allow some things to get in or get through and other things not to. That's because of that phospholipid bilayer. So I'm going to go to another page and just quickly describe the process and the concept of osmosis, as well as some key terminology. If I have, I'm going to be simple. I'm not going to draw all those little balloon heads because it takes too long. If I have a membrane, a plasma membrane, and it's semi-permeable, and I've got a water-based fluid on this side, and I've got a water-based fluid on this side. If I dissolve something or put some kind of a solute, you can even think of something simple like table salt, like sodium chloride or glucose. Let's just say glucose molecules. If I put those in the water solution on the right, and I put some on the left, but not in the same concentration. Water always wants to move toward the area where there's a higher concentration of solute, of particles. So we call this higher concentration side hypertonic. That means there's more things, particles, dissolved in this solution than on the other side. The other side is called hypotonic compared to, so we're just comparing the right from left. The right would be hypertonic compared to the left would be hypotonic compared to the right. Water or water-based fluids always want to move toward the hypertonic side. So in this scenario that I've just drawn, water will flow through this semi-permeable membrane toward this side. It's semi-permeable. That means that these glucose molecules can't get through. They can't move, but the water can. And so in this situation, picture this. I'm going to draw it again. If this is the ocean, and this membrane is your skin, so this is, I'm just going to say this is you, this is the inside of your body. When you wade into the ocean or swim in the ocean has a higher concentration. It's saltier than the inside of our bodies. And so when you're in the ocean, when you go into the ocean, you lose water from your body to that environment. You become dehydrated for the same reason that I described on the left. The ocean is hypertonic compared to the inside of your body. So that's a little bit, osmosis is just the diffusion of water. So we've explained how water diffuses, the process of osmosis in a living system. The last example I'm going to give you, so I'm just going to erase some of this business. And I believe this is an example in almost every A&P book that I've ever seen. If I have a jar or a beaker with a solution in it, and it's got dissolved particles, you can think sodium chloride, you can think glucose, doesn't matter. It's got dissolved particles in it. And I have a living cell. Let's make it a red blood cell, because this is a classic experiment. If I take a red blood cell, which also has some things in it, but it's not as, let's say it's not as concentrated as the solution below it in terms of solute. If I drop this red blood cell into this environment, just like your body in the ocean, it's going to lose water. So I'm going to draw this cell in here now. And because the cell was hypotonic compared to the hypertonic solution, water flows out, it loses fluid, and the cell shrinks. It does this, it shrivels and shrinks because it loses its fluid. just like the skin on your body feels all wrinkly after you've been in the ocean for a long time. This process, this of shrinking, is called crenation. The cell becomes crenate. That just means to shrink or lose fluid. Likewise, the opposite is also true. Let me erase my ocean. If I take a different beaker full of a water-based solution and I put some solute particles in it, but I drop a cell in here, let's just say this red blood cell has more particles inside of it than the solution it's going to go into. Now the cell is hypertonic compared to the solution, which is hypotonic. If I drop this in here, what happens is the cell takes on water because water always flows toward the hypertonic environment. So water in this case, let's make it hypertonic first. Water will flow into the cell, and that cell will swell, and eventually it will burst if it's not removed from that environment. So that explains how water moves through membranes and why it moves. It always moves toward the higher concentration of solute or something dissolved in the water. And these terms that I've used, hypertonic, hypotonic, and if the two sides of the membrane, the environments, are exactly the same, we call that isotonic. Those are just terms of tonicity. That's all they mean. All right, I've talked about active transport a little bit. Don't get hung up. What I want you to know about active transport, I'm looking at my book right now, and it goes into primary active transport and secondary active transport. Don't get hung up on the different types of active transport. I want you to know more that active transport requires energy, it requires a pump, and it pumps something against its concentration gradient. That's why it requires energy in order to do that. Next to your book, let me just clear this page and call up a new whiteboard. I'm just going to mention these vesicular transport. This word, endocytosis, just means the taking in of material at the plasma membrane by a vesicle. So, all right, what does all that business mean? If this is a cell in the human body, and let's just say this is a bacteria that's gotten into our system. Some of our cells have the ability to endocytose things. That means literally engulf them like this, and then take them in to what's called a vesicle. So endocytose just means to take within a cell. There are different types of endocytosis. So I'm just going to list those briefly. Phagocytosis. Phago just means to eat or ingest. So that's what I've drawn as my example, is phagocytosis. I think it's translated as something like cell that eats or cell eating. What happens is the cell just engulfs usually a foreign object and then packs it into some sort of a vesicle like this. And what it does with that thing depends on what it engulfed. If it's a foreign microbe, it usually engulfs it and starts to break it down and devour it. There's also something called pinocytosis, which is similar. Pinocytosis, I think it translates to cell drinking rather than eating, and that's just for fluid, taking in fluid. This one is taking in some sort of a microbe or solid. But they're the same basic process. It's engulfing and taking in to the cell itself. And the term exocytosis is just the opposite of endocytosis. It literally means to spit out of the cell. So if my cell has something it needs to get rid of, could be this thing. Some immune cells in our body engulf bacteria like I've drawn up here. Then they break that bacteria down, and they have the ability to sometimes spit out the pieces through exocytosis. Or sometimes, for example, a protein might be made inside of this cell. Here's a protein that just got manufactured. All proteins don't stay within the cells that they're manufactured in. Sometimes they export those proteins through a process called exocytosis. We're getting there, guys. I'm almost finished for the day. Going to try to go in the order of your chapter. This term, membrane potential. Due to the transport of certain things across a plasma membrane, this term, membrane potential, literally means the potential for energy along the plasma membrane due to separation of usually ions of some sort. That's sort of a mouthful. A quick, easy explanation of that is going back to our active transport, our sodium-potassium pump. So let me use that as an example since I've already described it to you. Here's our sodium-potassium pump. It literally uses ATP to keep pumping sodium out here and potassium in here. So what we are doing is we are separating sodium from potassium, and because sodium is much more positively charged than potassium, we've created a more positive environment out here than in here. The separation of these ions, and therefore this charge, sets up the potential for this membrane to do something, to have energy. That's what this terminology means. It literally means separating something along this membrane, like these two ions. And in doing so, we've created a polar environment. Just like a battery has a positive end and a negative end. I'm going to try to draw a little battery here. That has the potential to do that. It houses energy, potential for energy. And that happens to a lot of the membranes, our cellular plasma membranes throughout the body, is this membrane potential sets up this separation, this polar separation of charge, and gives it the ability. We say that this membrane now is polarized because of this separation of charge, and therefore has potential for doing something. We'll go into great detail with, when we talk about the nervous system with that, how a nerve cell can fire an electrical impulse. All right, I'm going to get to, before I cut it for the day, I'm going to get to what's in the cytoplasm, different organelles. And I'm just going to give you a quick run through of different organelles in the cell, in a typical generic cell, and their functions. And I'll try to go in order of your book. So we are in the cytoplasm now. We're off the plasma membrane. The cytoplasm refers to everything inside the plasma membrane and outside the nucleus. So the cytoplasm includes cytosol, which is liquid. By the way, that's about 75% to 90% water. So all the liquid inside of the cytoplasm is called cytosol, and then all of the organelles that are suspended in that cytosol doing things. So let's go through those organelles. We'll get as far as we can. You've probably heard of a mitochondria. That is an organelle found in many of our cells, not all, but most of our cells. It's a double layered, so it itself has a double layer membrane that conducts, or I'm going to say creates, synthesizes ATP molecules. So this organelle is really important to our cells because that's where our energy molecule comes from. How it works is through a process called cellular respiration. This is the process by which some sort of energy molecule, like glucose, for example, glucose combines with oxygen to form ATP. Here's our cell. I'm going to draw a big mitochondria. They're not this big, but I'm doing it so you can see. Here's it's a double membrane structure. It's got an outside membrane, an outer membrane, and an inner membrane. The whole reason we eat food and we breathe air is for this reason. Let's just say you've just eaten some pasta or whatever, a candy bar, and those carbohydrates are broken down and there's glucose in your bloodstream. When glucose gets transported across this membrane, it starts to get broken down and we produce 2 ATP molecules. That all occurs in the absence of oxygen. All right, 2 ATP molecules per 1 glucose molecule. Now, in the presence of oxygen, now we have oxygen in our bloodstream, and it diffuses out of our blood. And where does it go? It goes to the inner membrane of the mitochondria. This thing that started to get broken down right here into pieces, so I'm just going to write, let's just write 3C, 3C, so 3C plus 3C. When we start to break glucose, we get 2 ATP. But if we have oxygen present, then the products of this go into the mitochondria as well. And through a process called the citric acid cycle and the electron transport chain, I'm not going to go into those details, that's crazy complicated. What we get is 32 ATP molecules. So you can see what boost oxygen gives to the energy producing system. I like to think of it as this, and I'll say this again when we study the muscular system. We all can run for about 100 meters while holding our breath. We're using up a lot of ATP in that process, but we're not producing very many ATP. But you know, if you've ever gone for a run or if you've ever done some sort of physical activity, after about 30 seconds, you have to start breathing, and you start breathing heavily in order to maintain the demand the body is putting on you. This oxygen that comes in, you can see the magic that it works. It produces so much more energy. This is so much more efficient when we have oxygen. Hence the term, back here in the previous diagram, respiration means in the presence of oxygen. That's where that word comes from. So all of this, the mitochondria, is where oxygen combines with an energy source like glucose to produce a whole lot of ATP. Next organelle is a ribosome. I'll talk more about ribosomes on Thursday. These are small organelles, and they're made up of 2 subunits. The subunits are synthesized in a place called the nucleolus inside of the nucleus. So that's where they come from, ribosomes. The subunits kind of look something like this. There's a big one that sits on top of a smaller one, but this whole thing is extremely small. It's one of the smallest organelles in the cell. Here's its claim to fame. Ribosomes are literally the site of protein synthesis. It's where all proteins get assembled. And next organelle, I'll describe how that works on the protein synthesis bit on Thursday. Endoplasmic reticulum. It's just a membranous sac or a set of membranes. So let me get rid of my ribosome over here, and I'm going to draw a cell. If this is an entire cell, and this is the nucleus of the cell, the endoplasmic reticulum is just this folded membrane that sort of comes off of the nuclear envelope like this. So it's always associated with the nucleus in a way, physically. There's 2 different types of ER. There's rough ER, endoplasmic reticulum, and it's called rough because it just has ribosomes all over it. I'm going to write ribosomes on the surface. So a rough ER might look like this. It has these little tiny ribosomes all over it. That's where it gets its name. The surface looks rough. That's opposed to smooth ER. Oh, let me tell you the function of rough ER real quickly. Basically, this processes and begins to sort of package proteins, because these are the site of protein assembly, protein synthesis. All of these ribosomes on the endoplasmic reticulum then receive raw protein from the ribosomes and process and package them. Second type of ER is smooth because it lacks ribosomes. Smooth ER, I'm just going to write no ribosomes. So it's not really protein related in function. What smooth ER does is it synthesizes cholesterol, which our body needs, especially on our plasma membrane, synthesizes cholesterol and steroids, which come from cholesterol. Steroids are hormones like testosterone and estrogen. Some books call this the Golgi complex. Your book calls it the Golgi apparatus. If you hear me say complex, it's the same thing. Just depends on what book you're reading. This is also some kind of a weird membranous, flattened sac-like thing that has vesicles that come off of it. So it's like this tubular network. So I'm going to draw the end of a tube here. And in essence, proteins enter this, newly manufactured proteins can enter the Golgi apparatus and then travel through this processing plant where they also get packaged, and then oftentimes they get packaged into these vesicles for export. So it's sort of like a protein packaging and export station. Not all proteins stay in the same cell in which they were manufactured, which they were synthesized. Sometimes they get exported to somewhere else in the body. That's pronounced lysosomes, Lysosomes. I always think of these as a recycle center of the cell. These are like large vacuoles. They're big circular structures like this, oval-shaped structures. And what they do is they engulf, break down, and can recycle some parts of old, worn-out cell parts, meaning other organelles. So you know that nothing lives forever, including the things inside of our cells. And when things die or start to break down, lysosomes are actually the recycle center for most of the debris. They also, well, I'm not going to go into the specifics of how they eat things, and they're full, I'm just going to say this, they're full of what we call digestive enzymes. That's how the breaking down occurs. These digestive enzymes create a pH, an environment that's much more acidic than the rest of the cell. So inside of a lysosome, the pH is around 5, which is 100 times more acidic than out here in the cytoplasm, which is a pH of 7, because we increase the acidity 10 times with each number in pH. So from 7 to 6 is a 10-fold increase in acidity, and then from 6 to 5 is another 10-fold increase. So it's 100 times more acidic inside. Next is a peroxisome. It's similar to a lysosome. The reason it's called a peroxisome is because it actually produces hydrogen peroxide as a byproduct. But here's what it does. It detoxifies the cell. Basically, this peroxisome can neutralize free radicals. It oxidases, so using oxidase, so oxidizes toxins. And what that means is that it uses oxygen to break down things like, I'm going to write example, alcohol. If you consume alcohol, that's poisonous to your system, and it's poisonous to your cells. These things inside of our cells actually detoxify, break down that alcohol. Here's the catch though. In that process of detoxifying and oxidizing things, it produces hydrogen peroxide, which is poisonous to the cell as well. The good news is that hydrogen peroxide stays inside of the peroxisome and never really makes its way into the cytoplasm of the cell. And then hydrogen peroxide actually gets broken down inside of the peroxisome. All right, we're almost done, guys. Promise. Just 3 things I want you to know about the cytoskeleton. Just like the name implies, these are the structural elements, the physical structural elements of the cell. And they include these 3 protein-based, amino acid-based structures. Microfilaments, intermediate filaments, and microtubules. These are listed in the order of their size. So these microfilaments are the smallest, then intermediate filaments, and then microtubules. They all have slightly different functions. Microtubules tend to function in the process of mitosis or cellular replication. These two tend to be more structural in function and make up not only internal scaffolding inside of the cell, but they also, the term cilia are these little hair-like projections on the surface of the cell. They're not hairs in the strictest, they just look like hairs under a microscope. There's usually microfilaments running up into these little hair-like projections. They're just extensions of the plasma membrane, but under a microscope, they look like hairs, so we call them hair-like cilia. We don't have flagella in our cells in the human body, so I'm not even going to really go into that. The only cell is the male sperm cell, the sex cell, that has a flagellum on it. The difference between a flagellum and cilia is a flagellum is a long whip-like structure and it's singular. Cilia usually come, there's a flagellum, these are cilia. Structurally, they're the same. There's usually there's microfilaments and intermediate filaments giving that flagellum, just like in the cilia, some sort of structure. Let's go on to the nucleus and call it a day. We got so far today through all these organelles that on Thursday, I'm only going to have to talk about protein synthesis, and I'll take my time and it will be a short lecture. The nucleus of all cells contains the genetic material DNA, deoxyribonucleic acid. This is programmed software of the cell, which basically dictates all the functions in the human body. So all of our, I'm just going to a new page, all of our cells, except red blood cells pretty much, have nuclei in them, have a nucleus. Almost all cells only have one nucleus. Skeletal muscle cells are multinucleate. I'll tell you why when we get there. So here's the nucleus. Inside of this thing is DNA. That's our genetic material. I'm not going to go into a really hardcore lecture on DNA. You know that it's a double helix shape. It's made up of two chains of nucleotides strapped together by these bonds. Usually that DNA in the nucleus looks like this. It's just a big mess. It's a big ball of mess. And we call, when the DNA is in this state, we call it chromatin. And that's how our DNA looks like most of the time. It's only when the cell is getting ready to divide or replicate that DNA actually packs together into chromosomes, this really specific arrangement that looks like this. I'm just drawing a typical chromosome. That chromosomes are just this mess, chromatin, that has become organized and tightly coiled. And that occurs just prior to cellular division. So I'll write chromosome. A section, a piece of that DNA on a chromosome is a gene. So chromosomes have thousands of genes on them. So a small section, specific section of a chromosome that is the software for a specific protein, that's called a gene. Now, when this chromatin business is just arranged like a big bird's nest or ball of yarn, here's really what it looks like. There's nucleotide chains that wrap around proteins. Here's a protein. And I'm just going to wrap it. And I'm going to draw another protein. This is really how our DNA is arranged in that chromatin state. These proteins that the DNA wraps around are called histones. And they give this DNA some sort of structure. So if you hear the word or see the word chromatin, just think that's the state of DNA when the cell is not dividing, and that's most common. Chromosomes only occur, that's an arrangement of DNA that occurs when the cell is getting ready to replicate or divide. The term gene is just a section of a chromosome, specific section that is the software or dictates a specific protein to be made. Histones are structural proteins that the DNA strand wraps around. I didn't mention the nucleoli. That's inside of the nucleus. So I'm going to draw a spot inside of here. And we call this the nucleolus. I'm not going to get super specific. It's just the area within the nucleus where ribosomal RNA comes from. So in the nucleolus, it's a specific area that houses ribosomal RNA. That's because it's the software for making ribosome structures. So I'm just writing software for synthesizing ribosomes. I think I've gotten as far as I need to today, guys, so I'm going to cut my recording after I do that.